Delighted to be here and present this latest work. Crime in Sweden is rising, as it is in all over the world. I'll just talk about Sweden today because there is an idea that it is a peaceful country and without crime, and that we're all walking at night every day and singing songs and are happy. It used to be true. I don't know the part about night and singing song, but crime has been increasing over time. And Swedes don't like to hear that, but it's true. These are the data. This is the BRA, the National Institution of Statistics. And not only crime is increasing, all types of crimes, especially violent ones. The number of cases that can be closed and the crimes that are solved is very, very low. 95% of the crime in Sweden are going unsolved. This figure is very high. It's higher than Germany. It's higher than in the US. And as Swedes police know that they have a problem, it's not a secret. In 2008, the national police chief stated, it is often very little of value to work with when it comes to theft. There are no witnesses and victims often don't know when the crime occurred. There's really not much we can go on, which obviously makes it hard to solve crime. What does it mean? A couple of months ago, there was a break into my garage. People are breaking into the garage looking for winter tires, stuff like that they can sell. I reported the offense on the police website. Nobody came to take any evidence. I just paid for it from the insurance. Next event, a couple of weeks later, there was a breaking into the university. Okay, they broke in, they smashed the glass door. They came to look for chemicals, whatever. I expected the university would get more attention because it's university and it has budget and things to lose and everything. No one came again. That's what I mean by saying we don't see the police, and the chief of the police gives this has this attitude of we cannot solve it, so kind of looks like they don't even try to solve it anymore. So there is a problem, and I'm not putting it to of course to put anybody down. Of course not. We are working with the police. It's a problem everywhere. Crimes is increasing. Criminals are becoming sophisticated. I'm not even talking about digital crimes, AI and all. This is not my field. I'm talking about the regular offenses, the attacks on people's use of firearm, the use of explosive, all of those things. More traditional crime, they're all on the increase, and we need to keep up with this evolution of crime by offering new and better tools for law enforcement. With that respect, let me first pose a question: is there such thing as microbial forensics? Well, the answer is there is, at least on paper, and you can, if you search for it, you can find this expression being used at least far back as 10 years ago, at least as a concept. And if you find it earlier on, it's me using it. But in reality is that it is just a theory. It's just an idea. It is not being used in forensics. Although microbes are present in every crime scene and have been used, they're in every, they've existence and physical measurable presence. They've never been used in any case. Okay, and the question is why, right? If you have something that is everywhere, why is it not being used as forensics? Well, there are two possible answers. First, crime labs have historically been known to resist change. The police are not research institution like we're familiar with. They're not keen on wasting time on doing experiments. They don't have the time. They don't have the resources. They have this pile of cases unsolved on their desks. And it is not their interest to help us do our work that they think we need to do, or companies do. Yeah, answer number one. Answer number two, courts are slow in accepting new type of evidence. So even if we will go to the court and saying, hey, look, I've got microbiome evidence, they're like, what? Okay, and the police and the prosecutors will not go along with this scenario knowing that this is probably the answers they're going to get. And if you need more evidence, please consider that only in 1994, U.S. Congress passed the DNA Identification Act and CODIS, the national database of STRs for criminals. that is being used widely today. It happened only in 1994. The technology existed at least a decade earlier. Okay, so we're talking about very conservative, very resistant to change population. What does it take for microbiome to be court admissible? Well, the court is not resistant to change. That's important to remember. In the Dobert versus Merrill Doe pharmaceuticals case in the US, the Supreme Court outlined how court evidence should be evaluated in case of new evidence. They said it needs to satisfy all these 4 criteria. Was the tool tested? Well, as academic, the answer is usually yes. If we publish something, then it was tested. Was the theory or technique has been subjected to peer review and publication? Again, among most of us, the answer would, all of us should be right. The answer would be yes. It was peer review in a decent journal. It got some feedback. We corrected the criticism. We answered criticism. We published. It's good to go. What is known, the known or potential rate of error? Once again, if we did our job right, there will be some kind of confidence, some kind of measure, some kind of p-value that allows us to say I'm wrong in such and such cases, ratio. Has it reached general acceptance? A bit more difficult to measure, but I would say if one is being accepted to talks and their follow-up papers and the paper is being cited, one can argue that, yes, I satisfied that. Another point is that we don't have restrictions on collecting and analyzing microbiome evidence, okay? The collection of such data is unlikely to violate the Fourth Amendment in the US because once you're leaving something in the public domain, in benches or tables or coffee shops, one is allowed to collect it. And it's the same. The law does not say anything about microbiome, right? It's old, but it does say that about DNA. So there are no concerns for privacy. And if you're curious to read more about the legal aspect, I invite you to read my paper. In other words, we don't really have limitations on moving forward with the technology as far as the law is concerned. What is the limitation today? Very simple answer: the tools are not good enough, right? But papers are being published in forensic journals, usually consist of very small number of cases because of historical lack of budget in forensics. And when somebody publishes a paper with 3, 5, 10 samples, it's not a strong case. So what can we do to change the situation? Well, we can think about forensics application of microbiome. This is what I did. We can explore the legal aspects. This is what I did here. And we can dream. We can envision the future of how forensics investigation would be, and all the information that one can get from a dirty coffee cup, which is what I did here. Genome is in blue. I've been doing this before. I've been doing all those forensics, so working on ancestry, genomics. I can tell you which country you come from. In some cases, villages. Depends on the data we have. Appearance, profile, hair color, eye color. You know all those things. But where the microbiome is getting very, very strong is when you have to build social profile, right? Because unlike the DNA, it changes all the time, and it changes based on what it is that we do at the moment, what it is that we did last week, about our diet, all these things that the genome cannot tell us. And if you combine these two projects, you're getting something very, very powerful. And besides dream and review and vision, you can also collect a lot of data over a very long time. And this is what we've been doing as part of the MetaSUB project, founded by Chris Mason and colleague all over the world. In 2014, this paper, Cell paper, summarizes 2 years of data collected from subway stations all over the world. Every year, once a year, usually in the summer, PIs all over the world are going to the subway stations and swab. These data are being collected. It's being shotgun sequenced. And then the data were analyzed, and we found lots of bacteria, viruses, taxa, drug resistance, and a lot of other exciting taxa. The major findings were that outside of all this high diversity that you can expect from train stations, cities have core microbiome. So cities can be distinguished based on their microbial signature. And if you do classification, right, if I know the city and I know the bacteria and I can match them one to the other, we can get to 88% classification accuracy. Still not prediction, right? Prediction is the next step, but classification. And this was good enough for us to move on to the next step, which is prediction. And this is when we developed the microbial GPS, the geographic popular structure, mGPS. What mGPS does is true prediction. It is trained on samples, microbiome samples collected from water bodies, soil, and the cities of the MetaSUB project. There is some machine learning that is trained to find key taxa that are informative of location, and then it outputs the location on the map. All these data sets were generated by different projects, different technologies, different outputs. This is 16S RNA, this is shotgun, right? But the biological foundations are there. Bacteria are differences in different places, so the algorithm works. More specifically, I'm going to go into more details into what we did. We applied the supervised machine learning tool that employs geographically informative taxa. So this is a concept that I developed, and it differentiates taxa that are informative for geography from those that are not. And not all of them are informative. If a taxa is found everywhere, it is useless. These are the most geographically informative taxa. You can see this bacteria, for example, only exists in West Europe. This is extremely informative, right? If I'm finding a drug shipment that now, where the American boats are patrolling, right? And they're finding a ship with drugs and they're swabbing it and it comes with this bacteria, it did not come from South America, it came from West Europe. Okay, so this is why it's useful. And likewise, other taxa have also, this non-uniform distribution that if we take all of them, and there are 200 of those, right? And you put them one on top of the other, and you're looking at their relative sequence abundance, right? It's not just their whether it's there or not. We need to know the relative abundance, right? Then it is now possible to train some kind of machine learning on those abundances to predict the geographical origins of the samples. Okay. So 200 is the magic number. If you're curious, a small percentage of those were human pathogens. Most of these are not pathogens. They're associated with plants and specific bioclimate regions. This is what makes them unique, right? Because the climate in Australia is different from the climate elsewhere, so the bacteria will be different, right? So the first step, of course, before we do machine learning is feature selection. We need to find those GITs in a different environment to show that they're unique. What just happened? Yeah, if we look at all the data, it kind of looks like a blob, right? Different colors, different regions. But when we look at non-GITs, also means nothing, another control. But when you only look at bacteria that are geographically informative, all of a sudden you see clustering, right? So this is just a visualization to show you what the data we're working with. These plots don't go anywhere else. So after we found those GITs, we're going to train this machine learning, right? The data looks like this. These are the taxa on top. Samples are here from different geographical regions, and these are the abundance data, right? As usual, when we do machine learning, some data are being used for training, some data are used as unseen, and then we're training the model. Interestingly, in every model that we used, as urban environment, soil, or marine, it was always 200 GITs that were enough for the model to work. Okay. And then we used a chain model structure to make geographical predictions. So first we predict the continents and the countries and the latitude and the longitude. And this is the whole view of the model, just so you'll have it in front of you. This is the data collected from all over the world. This is what the relative sequence abundance of the data are. Then we're doing feature election, identifying the GITs, training the model, cross-validation, predicting all these geographical regions. And then once we have a model, then we're just giving it a sample, and it will tell us the geographical coordinates. How accurate it is? Super accurate. 92% of the samples were assigned to the sampling city. Station in Hong Kong was predicted 82% of the time. So this is when we had city-specific data. I'm really done. So in conclusion, we can make those predictions. We can go from taxa, from samples into geographical prediction, which is very valuable for forensics, because when you found those things on drugs and guns, you can tell where they come from. We're now collaborating with Malmo Police and other law enforcement to launch pilot studies to make this everyday use. Thank you so much.